Yes, the founding scientists and physicians have a deep understanding of the colon cancer field. We developed the initial molecular databases and have both discovered and developed diagnostic tests of some key genes in this field. An initial priority will be to develop a diagnostic blood test for early detection of colorectal cancer. Since a positive test result leads to a clearly actionable event, in other words, the patient goes for a colonoscopy. Now, the advantage of developing a blood screen is that it identifies a smaller pool of individuals who would most benefit from a colonoscopy. Yes, the fundamental technology will work for identifying other cancers directly from the blood. The National Cancer Institute has spent hundreds of millions of dollars supporting the Cancer Genome Anatomy Project, and this has generated copious amounts of data. Now, our team's data analysis approach can identify the critical characteristics required to identify and distinguish the different cancer types. Ultimately, the goal is to develop a complete cancer test. We will take a sample of blood and be able to detect breast, ovarian, pancreatic, prostate, lung, gastrointestinal cancers, including colon cancer, genitourinary cancers, brain cancers, head and neck cancers, melanoma, leukemias, and lymphomas. Now, such diagnostic screening tests would be performed as a two-step process, using an initial test to identify the presence of cancer in the blood, and then there would be a follow-up test to identify the tissue of origin as well as the best treatment. Yes, this is the future of medicine. Take a look at Eric Topol's book, The Creative Destruction of Medicine, How the Digital Revolution Will Create a Better Health Care. By analogy, in the 1920s, if a woman wanted to find out if she were pregnant, she went to her physician. Now, she just goes to the drugstore and buys an in-home pregnancy test. Likewise, diabetics can now continuously monitor their glucose levels with convenient in-home machines. In the future, versions of our blood-based cancer screening tests, the instrument will be dramatically reduced in size for direct consumer use and operated by a smart device. Now, to date, there have been 3 generations of drug development. The first generation is called small molecules, such as Tarceva. The second generation is called recombinant DNA-derived proteins, such as erythropoietin. And the third generation is known as monoclonal antibodies, such as Herceptin. Now, those last two, recombinant DNA-derived proteins and monoclonal antibodies, are known as biological drugs. Biologicals are too big to get inside cells, while small molecules are too small to disrupt protein interactions. Consequently, about 80% of protein targets are still undruggable. We have developed a new generation of drugs known as coferons. Coferons are orally active drugs that assemble on a target protein and interfere with or modulate the target protein's activity. Coferons can achieve extraordinary specificity with reduced side effects. And consequently, coferons may be used to target the undruggable proteins, which are about 80% of all the possible targets. Coferons now represent the 4th generation of drug development and may have a similarly extraordinary impact on treatment of diseases.